Hello, everyone. I'm Professor Jagath Rajapakse from Nanyang Technological University, Singapore. I'm attached to the Computer Science and Engineering Department, and my research is mainly application to computing algorithm for uh, uh, biological data and neuroimaging data. So today I'm going to talk about explainable AI and its application to drug discovery and neuroimaging. Now, the today's AI is mainly done by deep neural networks. So usually neural networks are considered as black box because they don't tell how the predictions are made, how the object detections are made. So they are considered as black box. But recently, there's an interesting to unravel this black box and giving some explanation to the process of prediction or process of object detection or regression problems that are performed by neural networks. Now, this has a lot of advantages because this will build trust with the users, because then users know why we are getting this kind of prediction or this kind of results from the AI methods. And also, as a designers, we will also get insight into how our model can be improved further. And also, uh, we will know the modeling, the input data or input data are processed or created by explaining our models of predictions. So this is the overview of my presentation. I'll give an introduction to explainable AI or XAI. And I'll introduce to you to the methods of XAI, the taxonomy, and then we will focus on one method, which is called the saliency backpropagation methods. And thereafter, I will explain two applications, one for drug mechanism prediction, and second one for neuroimaging. So this is how a neural network or AI method works. So you have some input, in this case, a brain image. So you have this black box or the deep neural network, and it will give you a prediction. For example, by looking at this image, it could be MRI, a CT, whether this person is healthy or not. That is what we are predicting using the AI technology or the neural networks. But so we can predict whether this person has a disease or not. But uh, the method, the neural network does not tell how it comes about this output, this outcome. So that is what explainable AI is going to do. It will take this prediction, and that method will explain how the outcome uh, resulted from this neural network. For example, it will find the regions that are different between healthy and unhealthy patients. Okay, so in order here we get these regions as the regions that are different from the patients and healthy controls. So these are called biomarkers. So sometimes this forward process of prediction is called encoding. So what we are trying to do is decoding the model to find out which inputs are most important. This is one of the objective, which inputs are actually creating the prediction. So these are actually simple classification of XAI methods. They can be broadly divided into two categories: input-based and activation-based. The input-based basically trying to identify which inputs are the most important inputs and how they are contributing to the output results. And the activation-based methods basically looking at the nodes activations and try to interpret the activations by looking at the maximally activated nodes. Okay? So we are focusing on the input-based methods, the backpropagation methods. They are the imports of the nodes. The output nodes is the most important node. So that saliency or the importance will be fed back using backpropagation to the input. So while you are doing the backpropagation, it will find the saliencies of all the nodes from output nodes to input nodes. Okay. And so there are two kinds of backpropagation methods. First one is the uh, uh, saliency backpropagation methods. That is what we are going to use today. And the other one is the class activation maps. Okay. And perturbation means you are giving some perturbation to the input individually and find out which input is causing the output to change most, so that you can identify which inputs are most salient or the key inputs, right? So these are the broad classification of XAI methods. But today we are focusing on the saliency backpropagation methods. That's what we are going to use for the applications. So saliency propagation method means that we know the importance of the output node because that gives output. So there is using backpropagation, we backpropagate that saliency of the node to the previous layer. And from that layer, we propagate to the previous layer, and we're going back to the input layer. So while we are moving to the input layer, we are giving some saliency or the importance of that node for the contribution to the output, so that we will find which nodes are actually creating this output, so that we can give some explanation. Okay, so there are different methods, slightly different methods coming under saliency propagation method: integrated gradients, layer-wise relevancy backpropagation or LRP, on deep lift. And here, your right-hand side, you will see the equations, the backpropagation equations, how the gradients are S stands for saliency, and the salience gradients are calculated and backpropagation from the output to the input layer, right? But I'm not going to go through all these equations, but they differ slightly different with the gradient equations that they are using to propagate the saliency to the layer before. So this is what we are going to use today to our applications. So these are some of the applications of XAI technologies. Healthcare, by applying XAI techniques, explaining the outputs, you can enlighten the disease mechanisms by revealing the decision-making process. And also you can build the trust, because when you give some explanation to your output, you can build the trust with the patients. Okay. And also in insurance, for example, by giving explanation how that prediction is made by the AI technology, you can build the trust, transparency, and auditability of the insurance in the insurance industry. And also in the manufacturing, by making you can use AI technologies to diagnose some mechanical faults in manufacturing industry and fixing equipment failure. But by making this process explainable, so you can create the awareness between the human and the machines, or the operators and the machines, so that they know how to use these machines better. And also autonomous vehicle, AI methods are driving these autonomous vehicles. So by providing some explanation to the methods, we can identify what causes accidents or how human can intervene to avoid these things. So those things can be sorted out by using explanatory methods of AI. So these are some of the applications. But today I'm going to do two applications that we have worked on. One is drug mechanism prediction. So drug means a chemical molecule, right? Usually a small molecule. So it has a chemical structure, right? It has a chemical structure. And this chemical structure is given to your body, and that will interact with some molecules, some proteins, some proteins, which are created by certain genes. So the drug mechanism means how this molecule or the drug interact with the biological molecules in our body, the proteins, for example. Okay, how the reaction. So this reaction can be predicted. Reactions can be identified by using explanatory AI. So this is how AI technique is using for finding novel drugs, for example. Okay, so a drug is a chemical compound, a small chemical compound. So this is the molecular structure. The structure is given by the chemical structure, and then genes and proteins are given by the genomic multi-omics data. So you can use genomics data to measure genomic expressions or mutations of the genes, and then gene expression data, proteomics data, and those data can be computed. So these data, the molecules in the body, proteins or RNA, such molecules will interact with the drug molecule, and then they make a reaction in the body so that it can fight the virus or the bacteria. So what we are trying to do is we are trying to decode this process and identify how which functional units of this drug compound is interacted with proteins or with genes. Because as you know, the genes are converted into proteins by translation and transcription and translation process. So basically, we can find which genes are actually interact with are affected by this drug. And if you know how they are interacting, then we can come up with novel drugs to certain diseases. So this is the process that we have worked on. We have worked on first the drug response prediction and using AI technology, neural networks. And then by using backpropagation, the saliency backpropagation, we have identified which genes are reacting with this drug molecule and which part of the molecule or which functional unit of the molecule are interacting with the genes. So we have found out. So using these findings, we can create new drugs. So this is the work that we are working in my lab. So I'm going to explain some part of it, how we use saliency backpropagation method to find the drug interacting mechanisms. All right, so this is a neural network that we have used to predict drug response from multi-omics data. So multi-omics data means transcriptomics data, which measure gene expressions. So using microarrays or RNA-seq data, we can calculate the expressions of thousands of genes. And then copy number variation data using genomic data, we can find the copy number variation throughout the genome. And then mutation data you can color RPA means protein expression data and the metabolomics data. So this data can be gathered today without much cost, and the cost is decreasing. So we believe in future With this, you can collect this multi-omics data to discover, detect diseases, and and uh, and identify biological functions. This multi-omics data can be used. For example, we have used cell line data and their expression data, their protein expressions, metabolomics data to predict how the cell react to different drugs. So we have used this AI, a deep neural network, to predict the drug response. So what we have is an input layer, and this is an embedding layer, and then we have a fully connected layer. And then we have a attention layer and an output layer, concatenation layer and the output layer. So there the saliencies are measured by the attention layer. With the attention layer, it will allocate weights depending on their importance for the drug prediction. That means which of which omics data are actually important for drug prediction. Okay, so here we have done this experiment. So you can see this is the decoding data on your right hand side. So you can see in this color map, you can see gene mutations and protein expression. RPPA data are actually more important in the predictions. So other data are not really important in predicting the drug response. So you can see actually these drugs are actually dealing with the mutations of the genome, as well as some protein expressions are affected by these drugs. So that is how we explain why we are able to predict drug response and which of the omics data are contributing to this prediction. This is another neural network. We call it XGDP. Basically, we have used drug chemical data. So it's like a graph, actually. If you look at the chemical structure of the drugs, it's like it has a graphical structure. So we have used the graph neural network, or GNN, to process this chemical structure of the drugs. And also, we have used gene expressions in this one. So we are trying to predict the drug using both gene expression data and the chemical structure of the drug. So by processing gene expression with the CNN and chemical structure of the drug with the GNN, we are able to predict the drug response. So here on the table, you will see the accuracies of our technology. So using different graph neural networks, and we find the GAT, graph attention networks, actually works well in this one. So so now we want to identify which genes are affected by this drug or controlled by this drug, and which chemical features of the drug is actually interact with these genes. So what we do is now we go from the output layer to the input layer by propagating the saliency backpropagation methods. So this the top, you can see the chemical structure of the drugs. So the color coded, it will tell you which part of the drug chemical structure is actually reacting with the gene expression data. So red means those regions are the most important regions that are interacting with the genes. And we have color coded the genes in the two bars. Actually, these vertical axes are all gene names. So you can see certain genes, the red genes are the yellow, the color bar is high. That means those are the genes actually affected by these drugs. Genes, those are affected by this. So we can give some explanations, not 100% explanation, but certain kind of explanation to this drug prediction, how these drug chemicals and genes are interacting with each other, and how they contribute to drug prediction. Which genes are actually acting with the drugs, okay, or which proteins? Genes mean produce proteins. So which proteins are actually interacting with this chemical structure, and which chemical structure are most important for this. So now we are working on actually by using this finding to create new drugs, drug candidates. So we have used some generative AI to generate new drugs using the interactions that we find from by decoding the drug response prediction. I'm not going to go into this. This is something that we are currently working on. So our next application is functional MRI data or functional neuroimaging. How the XAI can help us to identify biomarkers. Basically, we can have the connectome data, the functional MRI. We derive the functional connectome of the brain, and from DTI, diffusion tensor imaging data, we can generate the structural connectome. That means the brain networks in the macroscopic scale, and by encoding, we can predict certain things, whether this person has a disease or not, and then a disease subtype, the classification of the task the person is doing, and treatment response, cognitive measures, we can predict by encoding the imaging data. By decoding, that means we are going the backward. By explaining the important inputs, we can find the biomarkers. That means which part of the brain is actually different from, for example, different from healthy patients and healthy controls and patients. So we can intervene those regions by using a drug or some mechanism, those regions, to treat these persons. So basically, when you do decoding, we can find biomarkers, and also we can find the individual variability of the biomarkers. So this process, I will explain how neuroimaging is used in this expression. We are dealing with functional MRI data. The functional MRI will tell you which regions are activated during a particular task. But in this case, we are using resting state data of subjects. So even the resting states, some regions are activating in the resting state, right? So we build a network. We divide the region into certain, using a certain atlas, we divide it into regions, and then we build a network by connecting these regions. So basically, this network is represented by a matrix. That means the rows are actually brain regions and the columns are of the same regions. So in this matrix, one element represent how strong is the connection between one region and the other regions. So it's color coded here. The matrix is color coded. This is called the connectivity matrix, and this network is called the functional connectome. So connectivity matrix. So this is the matrix that we are going to use for as the input to our AI technique or deep neural networks. So by processing this deep neural network, say if you process healthy subject and patients, if you train this neural network using both kinds of subjects, when after training, when a new image is given, we can find the connectivity matrix from of that subject, and we can use the neural network to predict whether this person has a disease or not, right? So we can use the saliency backpropagation methods from the output to the input layer, so that we can find out which regions or which connections are most important. In the rightmost diagram, the connectivity matrix Now we have color coded. That means the yellow regions are the mostly important connections that create this differentiation between patients and healthy subjects. So in other words, you can use these regions or connections as biomarkers to intervene and treat these patients. Okay, so the biomarkers means we are trying to find out from the blue matrix, that's the resting state connectivity matrix, which connectivities are different from the patients and the healthy subjects. So those are called biomarkers, and those connections could be useful for treating these patients. So this is a network that we have used. We call it a lean. Basically, it's a feed-forward neural network. We give the connectivity metrics as the input, and the output will be whether it's a healthy subject or disease subject. So how we did that, this model we built, basically we did the forward pass by training this neural network, and then we use saliency backpropagation. So once we know the saliency backpropagation, we know in the neural network which nodes are the most important nodes. So what we did was we shed or we prune all these irrelevant nodes or the nodes which are saliency, low saliency. So we make a very small neural network by pruning the irrelevant nodes of this node. So we named this network lean, and we published this a year ago, this network. So using this network, since we found out the important, only we kept only the important nodes of the network, we got a very simple networks, as well as we could interpret the inputs, because only the remaining inputs are the most salient one for this prediction. So we have done this for using public domain data of different diseases. For example, the left hand side is the using AD subject and a healthy subject, and AD, Alzheimer's disease patients, biomarkers. So these in this color map, the yellow means or white means those are the most important connections that were different from Alzheimer's disease patients and the healthy subjects. So these are the biomarkers. On the right hand side, actually major depressive disorder, the patients and healthy patients we have compared, and these are the biomarkers that we identify using the previous network lean, that are different from this thing. And we have actually looked at the literature, and the literature supports our findings that actually these connections are actually affected in Alzheimer, or the right hand side, actually the major depressive disorder. Actually these connections are found earlier to be uh, uh, having issues confounded in these diseases. And then another technique, another neural network that we have developed is called SHRED. Because this neuroimaging data are very rare, so we have gathered these imaging data from different databases. But when you gather data from different databases, the subject's age, gender, demographic data are different. So we have to harmonize this data before we uh, before we harmonize to before we use data from different sites to use for prediction. So we developed a network which is called a SHRED. This is the network diagram you can see on the right hand side. It's basically it's a encoder, decoder, and classifier architecture, and we use the linear mixer model to pre-process the data and do this for the prediction. So by using this approach, we were able to uh, find site specific. That means now say if you get data from Singapore and we get some data from USA, what are the biomarkers that are invariant? Because the Caucasian population and Asian population, we can see which biomarkers are invariant using because we in our model using shred, we were able to combine data from multiple sites. So we what the biomarkers we get is from site invariant biomarkers. But if you train the network with only USA data, so those biomarkers are related to Caucasian population. So from the right hand side, we use a simple deep neural network that will give very site specific biomarkers. But on the right-hand side, we use the shred network, which will give site invariant biomarkers. We develop another neural network, which is called the split. So this neural network can identify subtypes of diseases. For example, we use the ADNI dataset to find the subtype of Alzheimer's disease because it's very heterogeneous. The AD patients are not very uniform. They have different phenotypes and genotypes. So we want to identify these subtypes. All right. So we build this neural network, which we call split. So basically, we have few graph convolution neural network layers, and once we get into a upper layer, we get a representations of the input data. Those are the connectivity matrices. And at a higher layer, we classify the cluster these activations. So these clusters corresponding to different subtypes of subjects, right? Subtype of subject. So we did, and we found that there are 4 subtypes of AD, and these AD subtypes we had the SNP data also, single nucleotide polymorphism data. So we could map which SNPs are corresponding to which subtype. So we could find the biomarkers corresponding to different subtypes. So the 4 subtypes, these are the biomarkers corresponding to the 4 subtypes. So with this, I have come to the end of my presentation. In summary, I introduced you to what explanatory AI means, what are the techniques, and then I explained the saliency backpropagation method, where the saliencies are backpropagated from the output to the input. And then I showed how the drug-target interactions are predicted by saliency backpropagation of using saliency backpropagation. We feed back the saliencies of the nodes, predicting the drug from the neural network, predicting the drug response backward, and find out the genes which are interacting with particular chemical features. So what we did was we had both gene expression data and the chemical structure data, and we do a graph neural networks and convolution neural network combination to predict the drug's response. And then we fed back to find out which drug functional units are interacting and which genes are affected by this drug. And thereafter, I explained an application to functional neuroimaging, where we use connectome matrices, functional connectome matrices from resting state fMRI data. And that connectome was the input to the neural network. And we use that to detect the different diseases from resting state connectome data. And we fed back using saliency backpropagation to find out biomarkers, which connections are actually different in these diseases and the healthy patients. which can be used to for interventions in future. But these explanatory variables have to use with some care, because there are some limitations of deep neural networks in building deep neural networks and so on. All right, so that's the end of my presentation. Thank you for your attention.